Welcome to ENT. Today's topic is about stem cells. Definition: it is an undifferentiated cell of a multicellular organism which is capable of giving rise to indefinitely more cells of the same type and from which certain other kinds of cells arise by differentiation. So this is a stem cell. It can give rise to muscle cell, fat cell, bone cell, blood cell, nervous cell, epithelial cell, immune cells, sex cells, etc. General background: stem cell types and properties. So the various types of stem cells we get to see are adult tissue-specific stem cells, embryonic stem cells, induced pluripotent stem cells, mesenchymal stem cells, and the umbilical cord stem cells. Let us know more about adult tissue-specific stem cells. It has been known for well over a century that many tissues of the body are continuously renewed by cell division. Detailed studies of the cell division in bone marrow indicated that many blood-forming cells have some ability to divide, but the ability to divide persistently, the key feature of stem cells, is possessed by only a subset of cells. Subsequent studies indicated that stem cells in the hematopoietic system can be identified by their expression of particular cell surface markers, which are totally clonogenic in vitro and have the ability to completely restore the hematopoietic system in mice whose bone marrow has been destroyed by irradiation. These findings led to the concept of an adult or somatic stem cell as an immature cell that is capable of dividing indefinitely, both to renew itself and to produce cells maturing for tissue function. Cells with such properties have now been identified in most normal and malignant tissues. So these are a few various locations of somatic stem cells in the body. It includes teeth, blood vessel, brain, skeletal muscle, peripheral blood, bone marrow, liver, gut, heart, skin, etc. Stems with the ability to generate many different types of cells are considered as multipotent. Other stem cells, for example those of the epidermis, normally produce cells of only a single lineage and are considered as unipotent. However, even cells that are normally unipotent may under some conditions such as wound healing be capable of generating cells of different lineages. So this is a morula. It is a totipotent cell which can give rise to embryonic stem cells which include inner mass cells and blastocysts. These are both pluripotent cells. So the embryonic stem cells give rise to multipotent hematopoietic stem cells which undergo differentiation to form RBC, WBCs and thrombocytes. Mesenchymal stem cells are also derived from embryonic stem cells which are multipotent cells which undergo osteogenesis, chondrogenesis, myogenesis, tendoligamentogenesis and others which undergo differentiation and maturation to give rise to bone, cartilage, muscle, tendon or ligament, marrow, fat, skin or other tissues. Uses: neural stem cell grafting is used for treatment in spinal cord injuries. Embryonic stem cell derived retinal cells are used for treatment of macular degeneration. Stem cell derived beta cells for treatment of type 1 diabetes. Embryonic stem cells. Controlled in vitro, differentiation of embryonic stem cells into a required cell type can be directed using signals that mimic the tissue interactions that guide emergence of regionally appropriate cell lineages during embryonic development. However, embryonic stem cells are generated by destruction of living human blastocysts. A strong opposition to the use of embryonic stem cells has been expressed on ethical background. So the sperm and the egg fertilize to form a zygote, which in turn forms a blastocyst. Blastocysts consist of inner cell mass and outer cell mass and cavity. Embryonic stem cells are derived from this blastocyst, which undergo differentiation to form ectoderm, endoderm, and mesoderm. Blastocyst turns into gastrula, which has an ectoderm, endoderm, and a mesoderm. This gives rise to fetus, which in turn turns into a developing human baby. So these somatic stem cells are derived from them, which undergo self-renewal and also undergo differentiation to form cell types restricted to organ source and any other cell types. More about induced pluripotent stem cells. Studies of embryonic stem cells led to the identification of genes associated with the maintenance of stemness, and it was found that induced expression of stem cell genes such as OCT4, SOX2, CMYC, and KLF4 in differentiated cells results in their acquisition of stem cell properties similar to those of embryonic stem cells. In addition to their value for clinical tissue replacement, induced pluripotent stem cells provide in vitro models of genetic diseases and for assays of drug effects. Mesenchymal stem cells. Mesenchymal stem cells are fibroblast-like, have good expansion and differentiation potentials, and also possess immune modulatory properties. Typically, the cells differentiated from mesenchymal stem cells are osteoblasts or chondrocytes, but greater plasticity with an ability to generate cells of non-mesenchymal lineages is suggested by the reports of their differentiation into neurons or hepatocytes. So these are mesenchymal stem cells. They can give rise to chondrocytes, myocytes, skin fibroblasts, astrocytes, stromal cells, adipocytes, tendon or ligament fibroblasts, and osteocytes. Now the last, umbilical cord stem cells. Umbilical cord blood collected at birth is a rich source of stem cells that can be used in research and in the clinic to treat diseases of the blood and immune system. Another type of cell that can also be collected from umbilical cord blood are mesenchymal stromal cells. So the mesenchymal stromal cells can give rise to liver cells, fat cells, cardiac cells, nerve cells, cartilage and bone cells, and muscle cells. These mesenchymal stromal cells are derived from the umbilical cord blood. So this is an umbilical cord. A 15 cm sample is taken. The umbilical cord artery is used in extraction of cord blood stem cells, and the umbilical cord vein is also used in the extraction of blood stem cells. The Wharton's jelly contains the mesenchymal stem cells. Cord blood is rich in stem cells that can morph into all sorts of blood cells, which can be used to treat diseases that harm the blood and immune system, such as leukemia and other certain cancers, sickle cell anemia, and some metabolic disorders. Studying how stem cells develop into heart muscle cells could provide clues about how we could induce heart muscle to repair itself after a heart attack. So, how can stem cells treat disease? Replace neurons damaged by spinal cord injury, stroke, Alzheimer's disease, Parkinson's disease, or other neurological problems. Produce insulin that could treat people with diabetes and heart muscles that could repair damage after a heart attack. Replace virtually any tissue or organ that is injured or diseased. The likelihood of the immune system rejecting a transplant of embryonic stem cell based tissue depends on the origin of that tissue. Stem cells generated through SCENT or iPS cell technology, on the other hand, are a perfect genetic match. Scientists have reported that cryogenically preserved cells have no expiration date, and frozen cord blood possibly can be stored indefinitely. Thank you.